Hello, good morning, teacher and actress name. My team is conformed por Aguillo, Verla Carra, Fernanda, Camargona Montserrat, Rodrigo Coquira Dame, and Bella Ledesma Lorena. Here's a bit about the insulin. Introduction. The, the insulin hormone secreted in the parietal gland is a hormone in the inlet carbonate lipid protein metabolites. The insulin secretion decreases insulin the action the parietal tissues. Insulin hormone are the broad cannot use the pass into cells. Decreases from the food and drinks. Just consume. His the body makes the circuit energy. The insulin plays the role is maintain the correct blood, cures the livers in the or choose the overload. It cures the serious heat problems. Argon cures the livers are known as hyperthermia. Means the blood clears liver are the ends and when the body does not produce insulin. It is not the insulin decrease the canal, the cell remedies the classroom. The hyperglycemia is instead blood decrease the limit are the low where the body send the more insulin in the blood. To most decrease the crux inter cells and delirious remains and is the progesterone. What is the function? The function is the primary function the insulin is the main blood to use the liver without the 18 and 100 distinct of the insulin. The educated cell the door is the had insulin raise the transport to use the various cell contributed so they make the fast cell. The cells they use the increase energy of the body. The function that transports the glucose is the energy body. In the cell, the constraint into energy, the process the glucolysis is used to control the blood sugar in the people. With the topic the one diabetes, the condition the is the bodies and not the make insulin and the body cannot the control the amount the sugar in the blood. In the people as handed the type the due the diabetes has the conditions with the blood sugar is in because the bodies does not make the or use the insulin normally. They cannot be corrected with oral medicaments. Honey, the human insulin is the class mediums they carry elements. The human insulin is used to take the place the insulin normally diffuse with the body. The words helping to move the blood in the other zones of the body. They reduce the energy. They also prevent the from the making the more the sugar. How is the topic in the insulin? How is the word in the white? The TB insulin dinner only is cranking, the shoulder working, and how the load continue the control the sugar.
I'm going to explain the importance of the insulin. The human insulin is used to control blood sugar in people who have type 1 diabetes and condition in which the body doesn't make insulin and therefore can't control the amount of sugar in the blood, or in the people who have type 2 diabetes, a condition in which blood sugar is too high because the body doesn't produce the insulin normally, that can't be controlled with oral medications alone. The human insulin is in class of drugs called hormones. The human insulin is used to take the place of insulin that the body normally produces. It works by helping move sugar from the bone or the other tissues in the body. Where it is used for energy, it also prevents the liver from producing more sugar. That is why the case of protein drugs, the pharmaceutical industry has opted for the path of genetic engineering, or recombination in the DNA metabology. By means of the technology, large quantities of a protein can be obtained completely from the cellular compounds of the original organism. This is accomplished by introducing a gene, can be host organism, for example, the bacteria. This organism is called genetically modified organism, and the obtained protein recombination in the protein. The importance in the metabolism, the insulin allows the entry of glucose into the cells, mainly muscle and fat, so that they have energy and the same time favors to the storage of glucose in the form of glycogen. In the liver and in the muscle, when enough glycogen has accumulated, the rest of the glucose by insulin is transformed into the fat that is stored and the fat cells. It has more actions among the main the importance in about the carbohydrates. This facilitates the trade of glucose into the cells to provide energy. For example, the glucose enters the brain cells without the need for insulin. It facilitates the deposition of glucose into the form of glucogen into the liver of muscles mainly, and it was involved in the transformations of excess glucose into fat and its accumulation in adiposities. The next is about the fats and proteins. These facilitate the formation of fats, for example, in the lipogenesis. They inhibits the breakdown of fats into facility acid lipolysis. The next is the facilities the transport of amino acids to cells that are the basis for the protein information.
How is the insulin made? The insulin is hormone that regulates the level of the glucose in the blood. Your cells require energy to carry on this process. Glucose is the main source of energy for the body. When obtained glucose for food, which through digestion is converted in nutrients, including glucose. The insulin is secreted when there is hyperglycemia. That is when there is a lot of the glucose in the blood. What usually happens after eating? Insulin causes the uptake, storing and consuming for glucose in also all through of the body, but specifically in muscle adipose tree of the leaf. 2. In the liver, insulin facility to entry or glucose into liver cells, prevent the receipt of glucose into the blood, promote cyclogen synthesis in the muscle, increases to the transport of glucose into the muscle cell when it can later be consumed for the energy from the ATP in adipose tissue. Insulin indirect promote the storing for fat in the front on the triglycerides. It is a typical experiment of a blowout protein in church when find the non-polar amino acid in the center forming cytophobic nucleic and the polar and charged amino acid around it. It is a G-conserved structure when specific. Sure is the case and the human insulin only difference on the amino acid for the pig insulin. The insulin obtained for the pig and was is also identified to the human insulin, and it comes because used efficiently by the body to convert carbohydrates in energy. Following this technique, in total more than 2,000 points for the extraction on 145 milliliters or purified insulin. Today, the process of the extract insulin for trouser or animals glands can be crossed both back in the was breakthrough and diagnostic of diabetes was as the sentence.
My name is Carla. I'm going to talk about insulin production. The general technique. Special enzymes are used to insert human gene into the DNA of bacteria. Human genes make new material which they were not able to produce before. Under the right condition, microbes multiply rapidly so that a large amount of the new substance can be generated in a short period of time. In this way, these genes that control the production of human insulin can be placed in a bacteria cell in order of the microbes to make human insulin. Later, this substance is extracted from the bacterial culture. The two technique is a recombinant method for the insulin production. First, the genes are cloned with the A-galactosidase in the plasmid PBR322. We can see the image that the recombination plasmids are the amplified in the E. coli transformation of the bacteria is carried out in two different nucleotides. Then the reaction between the GALAT protein and the insulin gene with methionine causes the protein complex to change. Thus explain the methionine molecule and eventually producing insulin with fewer amino acids that the beginning of the process by the recombination method. The mechanism of the separation of insulin is a production of insulin through the bacterial chromatography, which is used to separate insulin from other species, based on a wide variety of different amino acids. And thanks to the hydrophobicity of insulin, relay to another component is needed to achieve a proper function, the carry out certain steps. The first is the gene of interest tendency of the protein must be generated with must be the large protein in a phase stationary.
In conclusion, the medicine you take will vary according to your type of diabetes and whether the medicine controls or blood glucose levels and other factors such as additional health problems, medicine costs, and your daily activities can influence the medicine you take for diabetes.
Insulin is very important because it's the accumulation of the glycogen in the liver and fat the use. The earlier the body adds energy, the reduce the calories consumed during fasting, the time how to control the glucose level. The DNA and the recombination technique, the bacterial plasmid, is the part of the genetic energy. The technique that is the create the application of the scientific and economic interest. The application I have the experience the spectrum of the benefits. The not only medicine, also the manufacturing, the other number of the reduce the range from the production. It is the reference. Thank you for attention.